love to talk to mitochondrial transplantation in advanced engineered tissues to uncover mechanistic drivers, because I think for the success of our initiative, it is absolutely critical that we figure out the mechanism. That is the key. How does mitochondrial transplant work? Otherwise, I fear that we may suffer the same fate some cell therapy in some areas have suffered, where there was a lot of effort and no real success so far, for example, in cardiac cell therapy. And so my main focus is engineering heart tissues, and if we think about how to do that, like when I look at the heart muscle, right, I see elongated cardiomyocytes, they're all aligned in parallel, but I also see another structure there, and that's fractal vasculature. So as engineers, we are always trying to reproduce this structure, but how do you get different types of structure from different multi-cell types in the same tissue? So I'll just tell you one story on tissue vascularization that was pioneered by Shira Landau when she was in my lab. She's now a professor at Technion. So let's say I want to make a vasculature. Like, I definitely need endothelial cells. But if I try to just use endothelial cells, they cannot self-assemble and make stable vasculature by themselves. So I have to add something else. And the other ingredient that this is well known, it's not a novel finding from our lab, you need to add some stromal cells. And Shira, for example, used dental pulp stem cells, but you should probably use pericytes or fibroblasts that are found in that tissue. And because the stromal cells secrete a lot of angiogenic molecules as well as matrix molecules, they can promote self-assembly of the vasculature and stabilization. So here, HUVEC cells are labeled in green, and you can see that they are maintained over 2 weeks. So now we are thinking, okay, let's add a third cell type that I need in the heart muscle, and that would be the cardiomyocytes. So when I added the cardiomyocytes, when Shira did it, we can see that all the vasculature is gone, right? Now I add one more cell type, the vasculature is gone. And indeed, it looks like there is something that's secreted by cardiomyocytes. This is actually the result of supplementation of exosomes from cardiomyocytes to endothelial cells. You can see that in a scratch wound assay, they slow down cell migration and proliferation. And these are really, really important hallmarks, very important things for angiogenesis. There is also secretion of pro-inflammatory cytokines. Just by addition of additional cell type, like 3 cell types, you create a very, very inflammatory environment. And it doesn't really matter how you vary the ratio of the 3 cell types. It is almost impossible to have them coexist. If we have a lot of cardiomyocytes that are required for contraction, endothelial cells will sort away. And so, but we need both of them. So how do we solve this problem? And we live in a very unique time in the scientific history because we have single cell sequencing and single nucleus sequencing, and we can really peek into native tissues much better than we were ever able to do that before. And from single cell sequencing studies, obviously now we can see there are a lot of cardiomyocytes in the heart, but there are also other cells like endothelial cells, fibroblasts, and a lot of immune cells. And by far, one of the most important immune cell in the heart is primitive macrophage. And I learned a lot about primitive macrophages from Slava Appelman, who is the scientist here in Toronto General. So they arise from the yolk sac, and they seed all of our tissues during embryonic development. So every single tissue, almost everyone, will have macrophages, primitive macrophages. And they orchestrate homeostasis and vascularization, really tissue development during embryonic stages and later on. And even the ability of neonatal hearts to fully regenerate after injury is really governed by these primitive macrophages. Now, how do you get primitive macrophages? It's a little bit of a chicken and an egg. You need a primitive macrophage, and then they spend some time in the cardiac tissue, and then they become the resident macrophage. So you need like a tissue to program the resident fate, but the two things are connected, and how do you do that in vitro? So we were lucky to be working in close collaboration with Gordon Keller, and he figured out the protocols with his team of how to create primitive macrophages from iPS cells and human embryonic stem cells following this primitive hematopoiesis. And at the end, you get cells that look like this. They express LIVE1, and they are CCR2 negative. CCR2 is a marker of bone marrow-derived macrophages. So LIVE1 positive, CCR2 negative. They also express markers such as CD163, which is something that we often consider in bioengineering to be M2 macrophage marker. So when Shira and the team in the lab added this fourth cell type into co-culture, they were able to stabilize the vasculature over a long period of time in the tissues. So now we can see macrophages found really close to vascular cells, and on the cover of Cell Stem Cell, they show them as little construction workers building the vasculature of the heart. It was really important that the macrophages are really in close proximity to the cells, that they're really right there. If they're in the transwell, we get some effect, but it's not so pronounced. Importantly, this vasculature is perfusible, and we can measure and benchmark permeability, which in fact was very similar to native vanules. If you look at this picture right here, if we don't add macrophages to the mix, we don't have perfusible vasculature at all. The dye cannot enter the lumens. There are really no lumens there. These are just structures of cells that are kind of connected together, but there is no lumen. This structure is not perfusible. Importantly, also, macrophages enhance maturation of cardiomyocytes. And now you can see multinucleated cells in the presence of macrophages, really nice sarcomeres, higher contractile force. But what does that mean? You can see that very nicely in these movies. So these are fibrin domes that have 4 cell types versus 3 cell types. This movie is running. The movie here on the left is running, but you can't really see it. It's actually on your right, on my left. You can't really see any contraction because it's so weak. But when you add macrophages, you can see remarkable difference in contractility. In red here are labeled macrophages, and endothelial cells are labeled in green. You don't see stromal cells, but they're there. And you also see the very, very prominent contractility in this fibrin dome, as well as in the BioWire model. But how do macrophages work? And this is a big part of our collaboration with Anna. We found that they actually eat up free mitochondrial DNA. And by eating up this free mitochondrial DNA, they create a healthier environment for the tissue. They reduce the cell death, cardiac cell death markers like LDH, and at the same time, they enhance angiogenic markers. And Slava and his team showed in a sister paper that was published in Nature Cardiovascular Research that this act of macrophages, primitive macrophages, eating this free mitochondrial DNA from cardiomyocytes actually programs these primitive macrophages into very specific cardiac resident macrophage fate that is almost impossible to get in vitro in other approaches. And they're also very, very important for cardiac tissue homeostasis. So we did 2 things. We basically figured out by single nuclei sequencing what are the cell populations that we have in the 4 cell type tissue. And we also took the culture media, and so we performed proteomics, secretomics, and this was collaboration with Carolyn Cummins, to figure out what is actually made, what ligands are made and secreted in the culture media. So when you do those 2 things, you can really figure out receptor-ligand interactions because you know what's made. Now, if you look at this result here, you can see it looks like there are a lot more cell types than we put in, right? We put in, I'm telling you 4 cell types, but you can see a lot more going on here. So there are less mature and more mature cardiomyocytes. And then we also have macrophages here. We have stromal cells that we put in, but we also have fibroblasts, like cardiac fibroblasts. Where do they come from? We didn't put them in. They're actually results of contamination during differentiation cultures. It's almost impossible when you start from iPS cells to get 100% pure cultures. And so they come from that contamination part of differentiation. But they're very important because from this receptor-ligand interaction, we figured out that macrophages actually speak to stromal cells and fibroblasts via secreted factors and make these stromal cells and fibroblasts secrete more angiogenic factors like hepatocyte growth factor they increase the angiogenic score in the stromal cells, and that is how they promote vascularization of the heart tissue. So you really have 4 cell types talking together. So I was kind of really thinking about this concept of 4 cell type, and I went to a conference at the end of last year in Cold Spring Harbor, and Ruslan Medvedov was there. He's one of the most prominent immunologists from Yale, and he told this story. He showed this result. This is a result from his preprint that I'm showing. If you start from decellularized lung and you try to recellularize it with a single epithelial cell type, nothing happens. Then you try co-culture, there is a lot of matrix degradation. You try tree culture, right? Now you're putting epithelial cells, endothelial cells, stromal cells. You essentially get a fibrotic lung, but only when you put the 4 cell types, you essentially get a lung that's recellularized and indistinguishable from a native lung. This is all done with primary red cells. So if you look across many of our tissues, and definitely the results that Ori showed earlier are completely consistent with what we are observing. The 3 cell niche is not sufficient, right? In all of our tissues, you will see that there is one specialized cell. For example, for us, that would be cardiomyocyte. For Ori and his team, hepatocyte. But they're supported by stromal cells, by vasculature, and by resident macrophages. And without this 4 cell niche, it's very difficult to make a functional tissue. And you can see that across in the lung, in the brain, etc. So we thought that for our team, the most important question is to figure out how macrophage transplant influences this 4 cell type niche. So David, and the first thing is which cell type picks up the most, right? Like really quantify the macrophage. They quantified the mitochondrial transplant per cell type. And when David Bodenstein did these experiments comparing stromal cells, endothelial cells, and cardiomyocytes, it looked like that stromal cells are probably not picking as much as endothelial cells and cardiomyocytes. But then amongst the non-myocytes of this cardiac niche, which one picks up the most? And Didi Yurtel, who's a PhD student in my lab, performed these characterizations, and she found that basically, if you look at the scale, primitive macrophage, by far, and this is the dose response, right? Mitochondrial transplant, how many mitochondria did you put in? By far, essentially 100% of primitive macrophages will pick up mitochondria, versus at that dose, only about 10 to 15% of endothelial cells or stromal cells will pick up the mitochondria. And we believe these macrophages are by far the most important cell type that will govern the response of the tissue to mitochondrial transplant. And that's why David decided to actually use these 4 cell types in all of his ischemia reperfusion injury studies. And he found that if you transplant the mitochondria to healthy tissue, basically, after about 1 day, you cannot really pick up the signal from mitochondria anymore. They're all gone, but they persist for much, much longer in the IRI settings. So in the interest of time, I'll stop here, and I had some slides on automation, but perhaps I'll just show you one video here. So how are we going to do this in a scalable way? How are we going to grow many, many, many cell types, many tissues with 4 cell types to study these dose responses? And with a team in the Acceleration Consortium, there is this very adaptive robotic system where we pioneered visual servoing, and we can really control cell placement and labware placement from the meter to the micrometer scale to control these, create these very complex precise co-cultures in 384-well plates where you can see vasculature and very precise placements of organoids. So here's the video of how the robot works. Okay, well, I'm not sure why it's not. I might have to just give me a sec. It will. I'm not sure why I cannot run the video. That is kind of strange. Oh, here it is. Sorry, I just had to click twice. So you can see the completely aseptic enclosure, and in a footprint of a 384-well plate, the robot can pick and place organoids create basically vascularization first, vascularized bed first, and on top of this vascularized bed, it can pick and place essentially any tissue of interest at sub-millimeter accuracy. So you can see cardiac organoids in a background of endothelial cells. And we are using for this organobiotech plates that are perfusable, and each unit has 3 wells, and in the middle well, we can grow endothelial cells surrounded by basically any organoid that you want. And in this case, for example, it's brain organoid. So that is the platform that we plan to use in the future to scale up the studies of transplant. So I'd like just to thank the human organ mimicry team of the Acceleration Consortium, and to leave you with this summary that I believe macrophages are by far the most important responder to mitochondrial transplant. And maybe that's the reason mitochondrial transplant works in every tissue, is because there are macrophages everywhere. And as engineers, we're controlling structure to achieve function. And at the end, I'd just like to thank everyone on our team and our funding sources, and we'll take the questions.